In this episode of Brain Ponderings, it's my great pleasure to talk with Jeremy Nathans at Johns Hopkins University, where he's a professor of molecular biology, genetics, and neuroscience. He's also a Howard Hughes investigator, very prestigious and resource enabling thing to have. And also, importantly, he's a member of the National Academy of Sciences. And he's, his early work that really got his work noticed was he identified the genes that encode the pigments for color vision. But in general, he's interested in the eyes. He's an MD and a PhD. And so he knows a lot about the genetics of inherited eye disorders, and hopefully will incorporate that. He's also very interested in development. So that will weave into that. So Jeremy, welcome. And could you talk a little bit? So you were born in New York City, is that right?
Yes. I only spent 2 days there, I'm told by my parents.
Oh.
In New Jersey.
Oh, across the river.
Right.
Okay. And then were either of your parents scientists or MDs?
Yes, my father, Daniel Nathans, was an MD. He was interested in molecular biology, in fact, in the very early days, starting in the 1950s, and decided not to pursue clinical practice, but instead to go into research really at the dawn of molecular biology. And he came to Johns Hopkins in the early 1960s, recruited to be in the Department of Microbiology, and he stayed here for his entire career. In fact, he and I overlapped, and I am in the same department where he was a faculty member. So we were faculty colleagues in molecular biology and genetics for about a decade.
And did he work on infectious agents, I take it then, or not?
He started out working on bacterial viruses in the 1960s. And then in the late 60s, he switched to a virus called SV40, which is a small DNA tumor virus, causes tumors in animals. And he thought this would be an interesting model system to look at the origins of carcinogenesis. At that time, virtually nothing was known about the molecular basis of cancer. And so that was really a prescient switch, because shortly after he started working on SV40, his colleague Ham Smith, here, who was also in the microbiology department, discovered restriction enzymes, the enzymes that cut DNA at specific sites. And Ham gave my father a sample of that first enzyme that he had isolated, and my father used it to cleave the genome of SV40 and observe distinct restriction fragments. He was the first person to run a gel of DNA fragments and see that you could cut DNA specifically.
Wow.
And the two of them ended up sharing the Nobel Prize in the late 1970s.
Yeah, fundamental. I mean, a big advance in developing tools for that everybody uses now. That's studying genes.
Yeah, that was a big one.
And then, okay, so you were all geared up to, so you went to MIT as an undergraduate and majored in chemistry. Then you went to Stanford for MD, PhD.
Mm-hmm.
And so can you talk about your experience at Stanford?
Sure. I started out working in the laboratory of David Hogness. Well, I worked in his lab for my entire PhD, but I started out working there on Drosophila, which was what the lab was working on. This was the early days of molecular biology, gene isolation and gene sequencing. And for any PhD students who might be listening to this podcast, I think this story will be interesting. So I spent about a year on a project that clearly wasn't going anywhere. I thought it was going to be a thesis project, but we were trying to transform Drosophila with clone DNA, which turns out to be virtually impossible unless you use transposable elements, which were at that point unknown, not yet isolated. So after a year of working on what was clearly a project going nowhere, I was my mind was prepared for something else. I was basically looking around for a thesis project, and I just happened to hear a couple of lectures about vision. One from Lubert Stryer. He's well known as the author of a best-selling biochemistry textbook.
Yes.
And the other by Dennis Baylor, who was a neurophysiologist at Stanford. And both, as it turned out, were working on vision. They were working on photoreceptor cells and the mechanism by which light absorption is converted into an intracellular signal, ultimately an electrical signal. So I heard these two lectures, and they were fantastic. Lubert Stryer had just discovered that the light sensing receptor, rhodopsin, in the rod photoreceptors was a G protein coupled receptor. It activated an intracellular cascade that was essentially analogous to the way that hormones like adrenaline work. This was an incredible revelation. This was around 1980. And Dennis Baylor and his postdoc, Qingwei Yao, who's actually now here at Johns Hopkins as a faculty member, had just perfected a technique for recording from single photoreceptor cells with a sensitivity that allowed them to observe the cell detecting a single photon, the ultimate sensitivity. And that was incredibly beautiful also. So I literally ran to the library after hearing these lectures. This was before the internet, so if you wanted to read something, you had to go to the library. Ran to the library and started reading about vision. And because I had been working in David Hogness's lab, and Dave was a very outstanding molecular biologist, a real leader in the field in the world of gene structure and function. I was thinking in terms of genes. And so as I was reading about the visual system, it occurred to me that the new recombinant DNA technology, the ability to isolate genes and study their structure and function, had the potential to answer some very central questions in visual physiology. In particular, the question of what the structure was of the light sensing receptors. This was not known at the time. I mean, it was known that they existed, but we did not know what their structure was. We didn't know in detail how they worked. We had some general ideas from the photochemistry. They all had a chromophore. It's called 11-cis retinal, a vitamin A derivative. But the protein part, the business part in terms of passing along the signal, was largely terra incognita. And it occurred to me that the best way to study it wasn't to try to study the proteins directly. That was technically very difficult, but rather to get one's hands on their genes. And that would allow all kinds of experiments.
Yeah. And then, so at that time, had rhodopsin been, the gene been cloned and sequenced?
No, that's actually what I did as a first step. So the protein is very abundant in the retina because, just by way of background, rhodopsin is the sensor in rods. Those are the photoreceptors that mediate dim light vision, the kind you use on a moonless night. They're actually the majority of the photoreceptors, the vast majority in the human retina, as it is in most vertebrate retinas. And the cones are the ones that are used in bright light and also mediate color vision. We have 3 different kinds, and each one has a different kind of light sensor for different wavelengths of light. One in the long wave end of the spectrum, one in the middle ranges, and one in the short wave ranges. So the rhodopsin, because the rods are so abundant, and rhodopsin itself is abundant within the rods, rhodopsin had actually been purified, and the protein had been studied in some detail biochemically. Its sequence was not known when we began, although people were working on it, sequencing the protein. So we did sort of an end run in the sense that we just simply bypassed the protein completely and got our hands on the DNA and sequenced the DNA, which is much more straightforward.
Yeah, and then, so the different pigments, there's three blue, green, there's rhodopsin in the rods, then there's 3 other opsins for color vision?
Yeah, so they're often somewhat informally called the 3 other ones, blue, green, and red, or the colors that they most efficiently absorb. The psychologists don't like that. They prefer short, medium, and long wave, which are more generic names. But yes, those 3. Now those, because the cones are rare, those were known. They were known to exist based largely on behavioral studies. That is, if you test people in color vision tests you can infer that they must have three different kinds of cones. And if you do the tests with very precise wavelengths of light, for example, using either a grating or a tunable laser to create very precise stimuli, you can calculate what the absorbance spectra of those sensors must be. But that's the level at which they were understood at the time. There was really no molecular data on what their composition was.
And so the light hits the, rhodopsin or the other opsins, and then that causes some change in the, what is it, retinal, cis11 retinal?
Yes, exactly.
And then how is that signal transduced into an electrical impulse or reduction in the electrical impulse?
Yeah, good question. So retinal is a small molecule. It's got a molecular weight of maybe 500. It sits right in the middle of the receptor. So now that we know the structure of the receptors, actually we know the three-dimensional structures at this point from work from Chris Palczewski many decades later. They have 7 transmembrane segments. These are membrane-embedded proteins. And in the middle of that little palisade of transmembrane helices sits the retinal in a little binding pocket. And what light does is, as you mentioned, light changes the configuration of retinal. It photoisomerizes it from the cis configuration, about one of its double bonds, to trans. So that is sort of a little flip-flop, a molecular flip-flop. And the change in shape of the retinal drives a conformational change in the protein molecule around it. So if you're thinking like a pharmacologist, you can think of 11 cis retinal, the form that's there before the light absorbance, as an antagonist of the receptor. It keeps it in the off state.
Yeah.
And then light converts it into an agonist, a molecule that changes the receptor or pushes the receptor into its active conformation. And then once it's active, it catalyzes the activation of a G protein. So these are cytosolic but membrane-associated heterotrimeric proteins. They have a GDP bound in the inactive state, but that GDP is released and it's exchanged for a GTP in the active state. And that's the tag that the protein is active, and that's the thing, that exchange is the thing that's catalyzed by an active G protein coupled receptor. So in that respect, rhodopsin is not different from any of the other G protein coupled receptors in the body, of which there are hundreds. In the photoreceptor, without going into all the gory details, the cascade after that is a little bit different from other cells. So the G protein activates a phosphodiesterase, which degrades cyclic GMP, and the cyclic GMP levels are monitored by a membrane channel, which binds cyclic GMP in the open state, and then when cyclic G declines, the channel closes. So it's kind of a funny system. Most neurons open their channels.
Yeah.
You alluded to this, Mark. Open their channels when they get active or get activated. The photoreceptors actually close their channels when they get activated.
Yeah. So, yeah, that's really kind of a counterintuitive thing. That so in complete darkness. Wait a minute. In complete darkness, the photoreceptor cells are more depolarized.
The channels are open. There's a big
channels are open, and then when the light hits, the channels close, and you get kind of a hyperpolarization.
Exactly right.
So what happens then after that? So
just to follow up your thought, so in darkness, when the channels are open, the photoreceptor is constantly releasing neurotransmitter onto the second order neurons. And in light, when the channels close, neurotransmitter release declines. It's all backwards. It's very bizarre. And it's also energetically very expensive because the cell has to be constantly pumping out the ions that are emitted during the times when it's not being stimulated.
And so when you're sleeping, these photoreceptor cells are active, and there's Are there any cases where people, the cases where people have some sort of blindness, say, caused by an injury to an optic nerve or something? Has anyone looked at dreaming in those people and see if I wonder if there's any, any effect on that increased activity?
I don't know about dreaming, but I think the system basically it's been built this way for millions of years. All vertebrates use this system as far as we know. And from an information point of view, it's not any worse or better than the normal way of doing business. That is, having more neurotransmitter release when a cell is active is one way to send a signal, and having less is another way to send a signal.
Yeah.
Why the good Lord made it this way, I say that.
And the neurotransmitter used is glutamate, an excitatory transmitter.
Yeah.
And then what happens? What cells do the photoreceptor cells synapse upon? Where's the glutamate released onto?
Yeah, so the second order neurons are bipolar cells, the middle layer of the retina. They're also horizontal cells in that synaptic layer that modulate these signals. And then there are many different kinds of bipolar cells. This is a fascinating area of research. I'm not active in it, but I've followed it, where the signal is basically getting parsed and modified within the retina before it's sent to the brain. And one reason for that is that the number of axons, wires essentially, going down the optic nerve from the retina to the brain is quite limited relative to the number of photoreceptor cells. So there's something like 100 to 1,000 more photoreceptors than there are axons going down the optic nerve. If every pixel in the retina, basically every photoreceptor, had its own private channel to the brain, the optic nerve would be as thick as your thumb, and your eyeball wouldn't be able to move around very much in your head. So the optic nerve has to be thin and flexible for reasonable eye movements. And also, your brain doesn't want all that information. It's too much information. So what the retina is doing is basically boiling it down to just the essentials that your brain really needs.
Yeah. Yeah, interesting. And then, besides this, kind of opposite signaling pathway in terms of inhibiting, hyperpolarizing the photoreceptors rather than depolarizing, there's another kind of a weird thing with the organization of the retina eye with regards to light. That is, you'd think intuitively the photoreceptors would be the closest to the light, but it's the opposite, right? They're way in the back of the retina, not at the front.
Yeah, I think people have wondered about that over the years. I think the logic is probably that the photoreceptors need to be up against the back wall of the eye, because the layer just beyond them, called the retinal pigment epithelium, it's basically a layer of cells that take care of the photoreceptors. It's like the combined liver and kidney for the photoreceptors. They degrade waste products. They help turn over the part of the cell called the outer segment, where the transduction machinery is. And also, there's a blood supply behind the retinal pigment epithelium, which is a very large blood supply, very high flow, very dense vasculature, which by diffusion supplies all the things that the photoreceptors need. So for example, because they have this dark current, the current flow in the absence of light, they have a huge appetite for energy.
Yeah.
They need a lot of oxygen, and that means they need a lot of blood vessels.
Yeah.
And so having the blood vessels behind the retina looks like it's a good design, essentially.
And there's no blood vessels in the photoreceptor cell layer. Are there blood vessels in the other layers, bipolar retinal ganglion cell?
There are in our retinas and in most mammalian retinas. It's not a very dense vasculature, but it's there. There are some animals like rabbits and guinea pigs that actually don't have vasculature. in their retina. All the neurons in the retina, photoreceptors and everything else, get their nutrition by diffusion from this large vasculature that's just beyond the retina.
And so the thickness, the entire thickness of the retina then it's got to be really small, like less than a millimeter, right?
Well, yeah, yes, it's maybe a fifth of a millimeter,
Yeah.
So pretty thin.
And is it, when did you get interested in development? So you've done a lot of work on development of the vasculature. And is that what got you interested in development, the blood vessels, or was it something else?
I would say I've been interested in development ever since I was an undergraduate. I took a great developmental biology course, actually from Richard Hines, the fellow who discovered integrins. He was a professor, he still is a professor at MIT. When I got to Stanford, the lab that I was working in, David Hogness's lab, was working on development in Drosophila. So this was the days when homeobox genes were just getting discovered and transcription factors being discovered. And so the sort of the excitement that molecular biology brought to developmental biology was very much a part of my sort of scientific upbringing. Yes. And the retina is a beautiful structure. I mean, everything's beautiful in the body, but the retina is especially beautiful. And trying to figure out how it's built, I think, is a great scientific question, still largely unanswered.
And okay, this is a good place for you to talk about Frizzled and Wnt. And can you give just kind of an overview of these? It's kind of a complicated system of ligands and receptors that seem to be controlling development, pretty much most all tissues. So can you talk about that?
Sure. Yeah, it's a fascinating field. So the word Wnt, W-N-T, is actually a combination of wingless, that's the W, and that's a Drosophila gene, yeah, and int, I-N-T, that's the N-T part, for integration site. And where these came from is wingless was a Drosophila mutation discovered many decades ago, which, as the name implies, had a defect in wing development. Actually, it turns out when you look in detail, there's also defects in the embryo. And so it was one of a set of sort of core developmental genes. Clearly, it was controlling something fundamental about body form and development. And then independently of that, Roel Nusse, who's now a senior faculty member at Stanford in the late 70s, was a postdoctoral fellow with Harold Varmus at UCSF and was studying the integration of retroviruses in the mouse genome that were responsible for generation of cancer phenotypes. Breast cancer, I believe, was the one they were studying, mouse mammary tumors. And he found that they integrated non-randomly. Turns out that what they were doing is they were integrating into the genome and activating expression of the adjacent gene. And the adjacent gene, if it was an oncogene, would then cause the cell to become cancerous. And so they mapped the integration sites, and they found, when they looked adjacent to them, that one of them, I think the most prominent one, was actually homologous to the Drosophila gene wingless. So here are these two, totally different species, totally different sort of biological processes of embryonic development and wing formation versus cancer, and yet they were being controlled by members of the same superfamily of proteins. And it turns out these are signaling proteins. They're secreted by cells. Mammals have 19 of them, 19 different Wnt's. They're all homologous to each other. And we, together with Roel Nusse in the 1990s, discovered the receptors, the Frizzled proteins. They were actually first identified in Drosophila, but it wasn't known what they did. And we identified them in vertebrates, and actually sort of took a good guess that they were the receptors for the Wnt proteins, and turned out that that was a right guess. But we still, I would say, to this day, don't fully understand what the signaling pathway does in all of its manifestations. As Mark, as you mentioned, it's involved in many different tissues. Virtually every tissue, every organ uses Wnt signaling for something. and one combination of receptors or ligands or the other does one thing or another.
Okay, and then which specific? Well, why don't you talk? There's this canonical Wnt signaling pathway. So could you describe that? Like what happens downstream of the Wnt binding to the frizzled receptor?
Yeah, the pathway it's very odd. It's biochemically strange. When the receptor binds the ligand, and also this involves what we call a co-receptor. It's a protein called LRP5 or a close homologue called LRP6. When those three get together, they cause a protein complex in the cytoplasm, which is right next to the plasma membrane, to stop doing what it normally does. And what it normally does is it phosphorylates a protein called beta-catenin. And when beta-catenin gets phosphorylated, it gets degraded so that the phosphorylation event makes it a good substrate for ubiquitination, which is a modification that leads to its degradation. So it's hard to keep all this straight in your head, but if you block the phosphorylation, then you block the degradation of beta-catenin. And by blocking degradation, beta-catenin levels accumulate. And when beta-catenin accumulates, it ends up spilling over from the cytoplasm into the nucleus. And in the nucleus, it binds directly to a transcription factor called LEF or TCF. And that complex of beta-catenin plus LEF TCF activates or represses various genes. Now, that's bizarre.
Yeah.
But
yeah, and so you've looked at a lot of things related to this Wnt frizzled signaling pathway. Could you talk about, I guess, specifically your work on role in development of the vasculature?
Sure. So in the vascular system, and I'll just preface this by saying we got interested in this through sort of a series of happy accidents. We were working on Wnt and Frizzleds and making mouse knockout lines. So eliminating one by one each of the 10 Frizzleds. So there are 10 of these receptors, 19 ligands. And one of the 10 knockout lines, it happened to be Frizzled number 4, gave a mouse that had very aberrant vasculature in the retina. Now, just at that time, this was the early 2000s, a Canadian ophthalmologist, Johann Robitaille, found a family that had a retinal vascular defect. They had insufficient density of blood vessels in the retina. It's a large family, and this was inherited in a dominant fashion. And she collected DNA samples, blood samples, and then DNA from many family members and tracked down the location of the mutant gene responsible. And lo and behold, it was Frizzled 4, exactly the gene we were studying. And it turns out that this vascular deficiency that Robitaille was studying is just one of several kinds of vascular deficiencies you find in humans. So the most severe form is called Norrie disease, Norrie, named after the ophthalmologist who first described it about 100 years ago. And tragically, kids who have Norrie disease are born blind. So they have a very severe hypovascularization. That is insufficient vascularization of the retina. The retina is very intolerant of not having enough blood supply. And the retina basically is degraded and becomes scarred and non-functional as a result. And then there are a whole series of milder versions that, including the one that Robitaille was studying, that involve just maybe part of the retina, typically the peripheral retina that's affected. So this was sort of described. It was observational data in the ophthalmology literature, but no one really knew what the mechanism of any of this was. And so we had a close look at this, and it turned out that the Norrie disease gene had been isolated just a few years earlier, again, by tracking it down in families where this was inherited. And it turned out that it encodes what looks like a secreted signaling protein. It had a signal sequence at the front end, It was, in fact, a far-flung member of the TGF-beta superfamily. So this is one of the big families of signaling proteins that does many things in development, kind of like Wnt's.
Yeah.
There are dozens of family members. This was one of them. Wasn't clear what it did, but presumably it had a receptor and it acted through that receptor. And so, again, we took a good guess. There's something to be said for good guesses in biology. So we thought to ourselves, well, the frizzled protein, which gives this vascular defect, is a receptor. The Norrie disease protein, which is called Norrin, the Norrin protein is a ligand. It needs a receptor. So maybe the two go together as a ligand receptor pair. Now, that certainly seems reasonable. Actually, at the time, it seemed a little less reasonable because we had just shown that the frizzleds were the receptors for the Wnt's.
Right.
So they already had their ligands. They weren't looking for new ones. And furthermore, TGF-beta has absolutely no homology to Wnt's. They're totally different structurally. We now have the three-dimensional structures of both, and they're totally unrelated. But it turns out that guess was correct. Norrin, this Norrie disease protein, is in fact a high-affinity ligand for frizzled 4. Interestingly, it won't bind to any of the other frizzleds. So we've tested all 10 of them. It only binds frizzled 4. And when it does exactly what a Wnt does. It activates canonical Wnt signaling. It stabilizes beta-catenin. So it's this very beautiful example of convergent evolution where these two completely unrelated protein families have members that do exactly the same thing.
So the frizzled 4 is in the vascular endothelial cells? Is that right?
Exactly right.
And where is the Norrin coming from?
Mostly from the glial cells.
Oh.
So basically it's instructive signaling from glia to the vasculature to tell the vasculature how to grow.
I see. And so what, in general, neurons are always born before glial cells in brain development. You have the neural progenitor cells, they proliferate, cells differentiate, then kind of later on you get astrocytes coming from glial precursor cells. So there's already so it's the glial cells that are really secreting the neuron to promote the angiogenesis. So I guess at that early stage, the early stages is still pretty early stages of development. So in the mouse, they're well, humans in general, the brain development occurs more in utero relative to postnatal. So in the mouse, kind of what's the timing of development of the retina in relation to birth and the expression of, well, not the expression, the appearance of the glial cells and the expression of Norrin and frizzled 4?
Yeah, it's a little different from humans, because mice have many children with each gestation. They are born at a more immature stage compared to a human, where it's typically one or most two.
Yeah.
So they come out blind. Their retinas are very undeveloped, and they only open their eyes at about 2 weeks of age. So kittens, the same thing. And so a lot of the action is postnatal in a mouse. I think, except for that difference, it's probably about the same sequence of events as it is in humans. Because the retina is quite thin, it can get some of its nutrition by diffusion up until a point where it's fairly well developed. So the vascularization is somewhat on the late side in retinal development.
So I'm thinking about, gene therapy. And so in a mouse, you might, say you're going to do this like an experiment where you've got frizzled 4 knockout mice or Norrin mutant mice, and then you let the animal be born, then you inject some construct that, or I guess do gene therapy, express the gene in that case if it's a loss of function. But in humans, you'd have to do it in utero almost, right?
Yeah, yeah, and part of the problem is, yeah, exactly. This is happening fairly early in gestation in a human.
Yeah, I was thinking, these kids that are born blind because the vasculature doesn't develop properly.
That's going to be a very hard one to.
And are those dominantly inherited mutations or recessive?
It turns out it's on the X chromosome, so the individuals who are affected are the males.
Ah,
Women who are heterozygotes are normal.
yeah. Okay, interesting. You'd almost have to do some. I've thought about this a lot from like inherited Alzheimer's disease, Parkinson's disease, early onset disease, Huntington's, and and this is almost an ethical question, or it can be, genetic counseling, and then giving the couple who's one of them's an affected person, for example, with some inherited disease, they're carrying the mutant gene, they can decide whether or not they want to have children. If they may decide not to have children, but they could also decide, I want to have children and I want to ensure that none of them harbor the mutation, because there are ways to do in vitro fertilization and embryo selection. And I've thought, I think as a scientist, if it was me in that situation, and I knew I had the mutant gene at that time, I'd really think seriously about doing that.
Yeah, Norrie disease is very hard on the children and the family.
Yeah, yeah. Okay, so let's get back to the frizzled then. So what's actually happening? What genes are downstream of the Norrie and Frizzled that stimulate the angiogenesis?
Yeah, that's a really good question. I think we're not entirely sure. That's an area of active research in my lab and other labs. One thing we're pretty sure is that the Norrie signaling pathway is not a spatial instruction to the vasculature to grow this way or that way. And I say that because unlike guidance molecules, for example, vascular endothelial growth factor, VEGF, is that kind of instructive signal. So a source of VEGF will attract blood vessels to grow towards it. And so there's a spatial sort of information content there. But for example, if you have a mouse that is missing the Norrie protein, it's got the gene knocked out. If you simply express the protein from the cells of the lens at the front of the eye and basically flood the chamber of the eye with Norrie protein, that fully corrects the defect.
Ah.
So what we think is that Norrie signaling, canonical Wnt signaling, in the vascular endothelial cells basically puts them into the correct transcriptional state. So they have to have a bunch of proteins to do the angiogenesis, and then other signals tell them exactly where to make the blood vessels, how to grow, what density to grow, where to branch, and so on.
All right, and then there's a common, I guess one of the most common eye disorders is macular degeneration. And that involves a hyper growth of the blood vessels. They're growing too much, excessive angiogenesis. Can you talk a little bit about that? And is there any role for these signaling pathways you're studying in that disorder?
Yeah, that's a really interesting potential connection. I say potential because it's not proven that this pathway is or isn't a player, but we know that all the components of the Norrie pathway, the ligand, the receptor, the co-receptor, beta-catenin, everything remains expressed throughout the life of the organism. And so presumably signaling continues, and we have good evidence. I'm just going to make a slightly circuitous path in answering this question. we have good evidence both in the retina and the rest of the central nervous system, the brain and spinal cord, that signaling through this pathway is critical for maintaining the blood-brain barrier and the retina version of it, the blood-retina barrier. And this is a whole specialization of the retinal vessels that minimizes diffusion of small molecules from the blood space into the tissue. It's basically a way to protect the central nervous system from toxins that might be in the blood.
Yeah.
So having said that, that's good evidence that because we know that signal is required to maintain the blood-brain barrier. That is, if you stop the signaling at any time in the life of an animal, the barrier properties will be lost. That's good functional evidence that the signal is happening throughout the life of the animal, and so is potentially relevant to any disease process that might happen later in life. And Mark, you didn't mention this, but I'll just mention it now, that macular degeneration of the usual kind is seen in older folks.
Right.
So this is something you might get in your 60s or 70s or 80s. There are rare forms that are seen in young people, but those are much less common. So I think a number of people, including us, thinks that this pathway does have a potential role to play in aberrant blood vessel growth, as seen in macular degeneration. I should say it's also aberrant vessel growth is also seen in diabetes. So diabetic retinopathy is what the ophthalmologists call the situation where the blood vessels in the retina are not doing their job, basically. They're not sufficient for the retina's needs, and new blood vessels start to grow. And invariably, as in macular degeneration, the new vessels have various problems. They're leaky, for example. They leak liquid into the retina, and they tend to break, so you get actual bleeding in the retina. So we and others, I mean, and I'm including also biotech companies, are interested in pursuing the possibility that modulating the Norrie and Frizzled 4 pathway could be a therapy for these disorders.
Yeah, I could imagine, injecting an antibody to Norrie into the eye, into the eye, or I guess gene therapy, knocking down, somehow decreasing the amount of Norrie, right? Should, in theory, and then, catch these people early in the disease process.
Yeah, actually, this is, I think, an open question in the field, whether one wants more Norrie signaling or less Norrie signaling. So your logic is correct in the sense that if Norrie is pro-angiogenic in the adult context, then we want less of it.
Right.
On the other hand, if the leakage of the aberrant vessels is the major problem that we're trying to treat, and Norrie helps promote the barrier properties, then maybe we want more of it.
Yeah. And
it's sort of a scale here. It's hard to know which side one wants to press the thumb on.
And you have mouse models of hypervascularization?
Yes.
Okay, so you have the tools to test this.
We can test it in mice at least.
Yeah.
Hopefully it will extrapolate to people.
Okay, let's see. You mentioned VEGF. I remember this was a long time ago. I think even when I was postdoc, or back in the 80s, late 80s, there was work on developing angiogenesis inhibitors. I think a lot of the focus was on inhibiting VEGF. I think also I was working on fibroblast growth factor 2, FGF2, which has important roles in regulating neurite outgrowth and stress resistance. And there's a collaborator actually up at Harvard, Seth Finkelstein. I can't remember the name of the guy that's famous for his work on the angiogenesis inhibitor.
Judah Folkman.
Yeah, and he was at Harvard too, right?
Yep.
Yeah, and so we'd shown in cell cultures that FGF2 could protect neurons against cytotoxicity, against energy deprivation, kind of stroke-like conditions, oxygen, glucose deprivation. And then Seth Finkelstein actually got as far as a clinical trial of FGF2 in stroke patients. And he published it. I don't know what happened with that. It had promising results. Are there many clinical studies going on now with, are angiogenesis inhibitors routinely prescribed to people with macular degeneration? I guess it's kind of the bottom line question.
That actually was a success story. So Napoleon Ferrara at Genentech, who interestingly trained as a obstetrician and then went to Genentech to work on a protein called relaxin, which would help birth, then sort of switched his research to angiogenesis. He discovered vascular endothelial growth factor, or I should say sort of rediscovered it because it had been discovered earlier. I'm blanking on the name of the fellow. He was at Beth Israel Hospital. He discovered this protein as a secreted protein from tumor cells, which causes a decrease in vascular permeability.
Sure.
No, sorry, an increase, pardon me, an increase. He called it vascular permeability factor. It turns out to be the same as vascular endothelial growth factor, which Ferrara had isolated in a totally separate series of experiments at Genentech.
Oh, yeah. Now this is all coming back. So tumors are highly vascularized, so that the cancer cells secrete factors, maybe VEGF included, that promote the growth of the vessels into the cancer cells. So the cancer cells are really good at getting nutrients, right? Because they're highly vascularized. And of course, they need the nutrients for the cells to grow. So there's kind of this almost evolutionary competition between the cancer cells and the person's normal cells.
Yeah. So, and I also remember the name Dvorak was the fellow at Beth Israel Hospital. So it's ironic because Judah Folkman, who was a pediatric surgeon who had this idea of basically starving tumors for their blood vessel supply. That was the overarching concept.
Yeah.
And he was looking for angiogenic factors made by tumors that he could inhibit. Just a mile away, Dvorak actually found the critical factor, vascular endothelial growth factor, but he didn't realize that it was a growth factor. He was studying its pro-permeability properties. And then the team at Genentech, being a biotech company, they realized what Ferrara had discovered and how valuable it could be potentially as a target for inhibition for therapy. They made monoclonal antibodies to it. Yeah. And those antibodies, when injected intraocularly, will substantially inhibit the growth and typically cause the regression of these new blood vessels in macular degeneration and also in diabetic retinopathy. And conveniently, thanks to Dvorak's observations, that this same factor is also a pro-permeability factor. Those antibodies inhibit that aspect of its function. And so the vessels become less leaky. So it's beautiful. So they're less, it's anti-angiogenic and it's anti-leak.
Yeah.
It's exactly what the patient needs. So these are widely used in ophthalmology, and they're pretty effective.
And all right, so one thing that promotes angiogenesis is it kind of reduced oxygen levels, like what we call hypoxia, right? And how does that work? How does low oxygen, so there's a role for VEGF in that, or is this something else?
Yes, so virtually every cell in the body has within it a oxygen sensing system, and it's a transcription factor called HIF, hypoxia inducible factor, and it's a very beautiful piece of biochemistry. It has a proline within it which can get hydroxylated. That is, it can get modified with an oxygen, and when it does, it gets degraded. When there isn't enough oxygen, that modification reaction is slowed down and the transcription factor accumulates. And when it accumulates, it turns on genes. And one of the genes it turns on is VEGF.
Yeah.
Actually, it turns on a whole set of genes to make the cell survive the low oxygen environment. But it turns on VEGF, which the cell secretes, which then attracts the growth of new blood vessels. So it's this beautiful homeostatic system where if you need more blood vessels in a particular place in the body, though the tissue will arrange for those blood vessels to grow just where they're needed.
And is that the mechanism that's responsible, for exercise? If you exercise your muscles, right, that can stimulate angiogenesis, I think.
Yeah, that's exactly right. And it's the one that that tumors use. Tumors make VEGF because they're hypoxic. If they grow and they don't have enough oxygen because they don't have enough of a blood supply, they'll make VEGF.
Let's go back to I want to finish up by going back to your earliest work again on the color pigments, the opsins. And I didn't ask you about color blindness. Essentially, you're identifying those, you could explain, and then looking at the genes in people who are colorblind versus those who aren't. You figured that out. How does that work?
Yeah, so color blindness is kind of a catch-all phrase that really encompasses a whole series of variations in color vision. The most common kind are a reduction from 3 different cone photoreceptors to 2. So the people who are, quote, colorblind in that way, they're not truly colorblind. They just have a reduced color world. Instead of essentially 3 dimensions, you can think of the activation of each of the cone photoreceptors as something you can diagram in a 3-dimensional space. So one axis is the long wave receptor, the second axis is the medium wave receptor, and the third axis is the short wave receptor. And so every light stimulus, whatever wavelengths or intensities it is, it'll be some point in that space. Actually, James Clerk Maxwell of Maxwell's equations was the one who first thought of that. And someone who is missing one of the 3 receptors, instead of having a 3-dimensional color space, a volume, they have only 2 dimensions. So it's a plane. And all the stimuli are points on that plane. Now, the reason we especially think about color blindness as corresponding to that kind of variation is because 2 of the 3 receptor genes are on the X chromosome. And when either of those is lost, the person, if the person's a male, they'll have only whatever remains, since males have only 1 X chromosome, an X and a Y. So it's color blindness of that kind is common in males simply because they have only 1 X chromosome. Blue color blindness, loss of blue sensitivity, or the shortwave sensor, is much rarer, and that's on a autosome, one of the regular chromosomes.
I see. And I also read some of your work, kind of an evolutionary perspective on evolution of these color opsins, color pigments. And there's kind of this interesting story as you go up and through the non-human primates and then get up to humans. Could you briefly talk about that? Because I thought that was pretty interesting.
Yeah, that is a fascinating story. Jerry Jacobs, who's a primatologist, he studies South American monkeys, is really the hero of that story. So Jerry, and I'll tell it the way Jerry found it. He was studying South American monkeys, like squirrel monkeys, and he made a very surprising observation, which was that the males are always colorblind in just the way humans are. That is, they're missing one of the 3 sensors. They have just 2 sensors, the shortwave sensor and then either a red or a green. Or actually a third possibility, they have a receptor that's sort of in between red and green. But in any case, they only have 2. But then what he observed is that females were often, but not always, trichromatic, not unlike us. That is, they had 3 sensors, but sometimes they had only 2. So this all sounds very confusing. But it turns out Jerry figured this out. And what is going on in squirrel monkeys is that they have just a single X chromosome gene, instead of the two different ones that we have. But the gene comes in 3 different flavors in the population. So it can either encode a longer wave sensitive pigment, or a medium wave sensitive pigment, or a pigment that's about halfway in between. Now, males, since they have only one X chromosome, basically get to choose one of those 3. They'll have one of those 3, and they'll, of course, also have a shortwave sensor, and so they have a total of 2 color sensors. But if a female, since females have 2 X chromosomes, if a female happens to be fortunate and get 2 X chromosomes that come with 2 different flavors of receptors, let's say one chromosome codes for a long wave sensor and the other one codes for a medium wave sensor, then that female will have in her retinas both kinds of sensors. And it turns out I'll tell you one more story, which is that in mammals, there's a process called X chromosome inactivation, in which females have a mosaic arrangement in which each cell in the female body expresses only one of the 2 X chromosomes. It's either the one that came from mom or it's the one that came from dad. But whichever one it is, that's the one that's expressed. The other one's turned off. And the decision to express one or the other is random, and it occurs early in development. So in these monkeys, these South American monkeys, who have 2 different kinds of X chromosomes with respect to their light sensors, some of the cells in the retina will express one. Some of the cone photoreceptors will express one of them, and the others will express the other one. And that means that the retina actually has within it, if you sum over all the cells, it has all the 3 different kinds of cones that we have. And so that female turns out to have very good trichromatic color vision. So we think that's actually sort of the path. That's the intermediate step on the way to the kind of color vision that we have. And then primates like us and chimpanzees and gorillas and macaques, primates that evolved in Africa, turns out we all share this gene duplication event, which gave us these 2 different kinds of genes on each X chromosome. That's, should we say, the most sophisticated kind of color vision that primates can have.
And so it's thought then that, so these monkeys in Africa, they're mainly living in our arboreal canopy and probably eating fruit a lot. And of course, as fruit ripens, right, changes color, usually to a bright color like a red or a yellow. And so it would seem to be a big advantage to be able to distinguish, all the colors, right, in terms of being successful in foraging.
I agree. Yeah, I think that was probably the evolutionary driver for trichromatic color vision, was to distinguish ripeness of fruits, green from yellow, from orange from red. And that's exactly what those X chromosome proteins do.
And also there's some thinking along the same lines that the evolution of the growth in the size of the prefrontal cortex, which is involved in decision making. There's been a lot of work actually gone on looking at foraging behaviors and the role of the prefrontal cortex in making choices to leave one area and go to another. So there may be some role in these color, and maybe indirectly in a way, a link between the evolution of the genes encoding the color pigments and whatever genes are involved in prefrontal cortex and decision making, because there's this interplay. So
yeah, and if you look at primates, the visual areas are also expanded. So there's this kind of parallel evolution of the primate brain where these 2 areas, the back, the occipital cortex, the visual areas, and the frontal cortex, are both sort of expanding dramatically relative to other mammals.
Yeah, that's fascinating. Okay, Jeremy, I appreciate you taking time for this. I've learned a lot, and I hope our listeners and viewers have too. And I'll put in the description section links to some of your papers, your lab page, and maybe some other resources.
Mark, thank you very much. This was delightful. I really appreciate your doing these podcasts.
Okay, have a good weekend.
You too. Stay
Bye.
well. Bye-bye.